We have everybody. Awesome. This is Dr. Jay Brock. He is our Director of Clinical Pathology here, so he oversees all the clinical portion of the labs, and he's our resident expert in molecular diagnostics. So he's going to talk to you today about all the fun and exciting emerging testing and give you a little bit of background about next-gen sequencing and that kind of stuff. So, Dr. Brock, thank you for being here.
Yeah, thanks for having me.
I have an extra water for you if you decide you want it. Thank you.
Thank you. Yeah, so I understand the purpose of this course is to sort of prep you for the exam. And some of the material that I'm going to talk about today is probably a little bit too sophisticated for like what you'll see on an exam, but you will start to see some basic questions on DNA sequencing and the types of techniques that we're currently using in the laboratory. So it's a little rich in content, and I'll give you a heads up for that. But don't, please ask questions. I'll send you the slides. You'll have this information. It's really good stuff to know because it's really what we're doing in the lab to bring better testing to our patients and improve patient care. And a lot of the initiatives at Akron Children's Hospital are using the techniques that I'm going to talk about today. So if you're planning on getting into this line of work and you want to do this as a career, these are the types of cool stuff that we're doing in the lab that are sort of cutting edge. This lecture was supposed to come last when you already had sort of all the background and the basic understanding, but I'll cover some of the basics. And if you have any questions, just sort of let me know throughout the presentation. So the topic is genomic medicine and testing. And what I really wanted to do, again, was sort of leave you with something that is sort of like, wow, this is really interesting. It's amazing what we can do for patients as it relates to looking at genetic information and giving us information about diagnosis, prognosis, how we treat patients, if they qualify for clinical trials, things like that. So that's the type of stuff that we'll be talking about today. Apparently this doesn't advance forward. So the first thing here is just like a basic definition of genomic medicine. So this is on the National Human Genome Research Institute webpage. It basically just says that genomic medicine is an emerging discipline. It involves using genomic information about an individual as part of their clinical care. So how do we look at DNA and determine how to best care for a particular patient? This is especially important in the pediatric world because kids have a lot of genetic disorders. In adults, you see a lot more cancer and heart conditions and things like that. But with kids, there's a lot of genetic constitutional disorders that are the etiology behind the disease and gives us information about how to care for those patients. Genomic medicine also deals with the policies and how we use this data and apply it to treat patients and making sure that we're doing it in a safe and responsible fashion. So that kind of summed up in this definition statement. And again, you'll have these slides, so you'll have this. Did you already get the PDF version? Okay. PDF version is pretty good. It just doesn't have some of the animation that you'll see in slides. So we're going to do like, again, this is meant to be more fun than intense. A lot of this stuff probably is too advanced for the exam, but you will see some questions. So it's important to understand and appreciate this stuff. But really, I want to try to have a little bit of fun with the lecture. So we'll do a bit of a timeline in how genomics has gotten to where it is today. The two key techniques that we'll talk about is next generation sequencing and optical genome mapping. We're starting to use these in our laboratory here at Akron Children's. We have a big genomics business plan, and we're going to bring on these technologies in the next year or two. So if you end up working here, then it's likely that you'll get involved with this type of technology. So then we'll talk a little bit about the application of this. So how do we use it for cancer? How do we use it to look at genetic disorders? Pharmacogenomics is kind of an emerging science where, based on someone's gene makeup, you can determine how they metabolize, and that helps in how we prescribe, certain medicine to patients, making sure that they get the right dosage and things like that. And then also it can be used for infectious disease. So we can use it for things like viral genotyping. Talk a little bit about COVID and how we sequence SARS-CoV-2, and that tells us about variants and circulating variants in the population. So these types of technologies. And then we'll do a brief quiz together, but it's not a graded thing, just for fun. So this all started in 1953 when Watson and Crick figured out the structure of DNA. And that was really important because in order to design tests that look at DNA, you have to first kind of understand how it's structured and how we can copy it and look at it. So in 1953, these guys came out with the structure, and it turns out that they were correct. And to this day, we still follow that model. So PCR was described in 1985. PCR is the biggest technique that we use in a molecular lab. And it's basically the process of copying DNA. So you start with the genomic DNA, and this technology enables you to make a bunch of copies of it. And if you have a bunch of copies, then you can see it with fluorescent techniques and microscopy. So PCR was a really big sort of thing that happened. In 2000, we finally sequenced the human genome. So at that point, we knew all the genes, all the content that was in every single individual. Humans have anywhere from 30 to 50,000 genes. The reason that it's such a broad range is that there's a lot of genes that are just involved in things like regulation and not necessarily protein coding. So you might see in your book or in literature that we have about 20,000 genes. Those are the genes that are really like protein coding genes that go on and have functionality. But really, all of the genes combined, we have more in the range of like 30,000. That's not something you would be tested on, but just sort of understanding that. In 2008, we came out with laws and statutes to protect us against this type of information. Because if you think about it, your DNA is really your fingerprint. Everyone has their own sort of set of DNA that describes you as an individual. So it's really important that we have protection in regulatory laws and statutes around how we use this information. So in 2014, we were able to get this down to like $1,000 per test. And that's really significant because in 2000, it took like $10 million to sequence the genome. Obviously, you can't perform a test that costs $10 million in the laboratory. So once we got this down to like that $1,000 range, that's when we were like, okay, well, we can use this in the clinical lab to treat patients and care for patients. And in 2016, the Precision Medicine Initiative started. Barack Obama was in office, and he signed basically an initiative by the government to support this type of testing for patients. So what have we done for the last 20 years? We figured out the genome in 2010, or 2000, sorry, and it's 2023. So what have we been up to? And I like this picture because it's a good sort of representation of what we were dealing with. When we sequenced the genome, we had this like gigantic undone puzzle where you have 3 billion puzzle pieces, but it's like, okay, how do we put these together and make sense of it? And that's what we've been doing for the last 20 years. We've been trying to figure out how to interpret this information and apply it in a clinical setting. So the first major tool that I'm going to talk about is next generation sequencing. Before I go into next generation sequencing, I'm going to talk a little about the basics of sequencing, understanding that this is your first exposure to some of this stuff. And again, if it gets a little rich, just stop me or ask me questions, and we can take time to talk through some of these concepts. So this is a very basic slide, and it kind of just breaks down like what is sequencing. So if you think about your genes are like books in a library. So a library has a whole bunch of books. So just imagine there's 20,000 books in this library. And sequencing is the process of reading all of those books. So making sense of what all those books say, what does that tell us about an individual? What are the clinical implications of that information? There's really two types of sequencing. There's Sanger sequencing and next generation sequencing. This is going to be on the quiz at the end. So this is a concept that might be on a test. Sanger sequencing is really looking at one gene. So if you think about this in the most simple, basic format, you're copying one gene and you're sequencing one gene at a time. And that was Sanger sequencing. And that's what we did for about 15 years until we figured out we can sequence a whole bunch of these genes at the same time. And next generation sequencing was initially called massively parallel sequencing, because the concept of it was we can sequence all the genes at once. So in a very basic sort of rudimentary depiction of this, would just be, sequencing all of these genes at the same time. So that's the main difference between Sanger sequencing and next generation sequencing, one gene versus many genes. And that's really the core concept. So why can we sequence DNA? So some basics about DNA. It's made up of 4 bases. So we have about 3 billion bases that make up our genome. And all of those bases are either an adenine, a guanine, a cytosine, or a thymine. So you'll hear people just say A, G, C, or T, and that's the code of DNA. And then depending on the sequence of that code, it tells us about genes and their functionality and information about an individual. So DNA is double-stranded, and that was really important because we realized that we could break apart those strands and we could copy a strand. Each of those strands are held together by hydrogen bonds. So A's always pair with T and G always pairs with C. So the concept of being able to copy DNA and sequence really came from that we can separate the 2 strands of DNA and we can copy one of them, and very similar to how DNA replicates biologically in the cell. So this is the process of DNA replication that happens in the cell. And you're going to get this in another lecture. I think Cassie Bonbasuto is going to lecture about the structure of nucleotides and how DNA is replicated and copied. So you'll see this again, but just as an introduction to this, as we go into the more complicated concepts of sequencing, what happens in the cell is you have this chromosomal packaging of DNA that gets unraveled, and these strands get separated by some enzymes that separate the strands apart. And then you have this primering event where an enzyme called polymerase comes in and copies the DNA strands. So in a sense, what you're doing is you're taking one copy of DNA and you're making 2 copies. And in molecular biology, in the process of PCR, it's just a series of steps where we copy the DNA. So you go from 1 to 2 to 4 to 8 to 16 to 32. It's an exponential process. And that enables us to design testing where we copy DNA and we look at gene information. So the concept of sequencing really came from this because we realized that we could do this synthetically in the laboratory. We could actually take DNA from an individual and use DNA polymerases, synthetic primers, and nucleotides to copy the template DNA and make a bunch of copies so that we could look at it in the lab and determine what's going on with the gene content in that particular individual. Does this concept seem pretty clear, straightforward? So how does DNA sequencing work? Again, this is a human eukaryotic cell, so everything's packaged in a cell. Within the nucleus, you have these chromosomes. We have 23 chromosomes that are copied. So in well, there's 46, but it's 23 copies. You get one from your mom, one from your dad. That information resides in the nucleus of the cell. And how that works is it's packaged into these chromosomes that ultimately unwind into this double-stranded DNA that can be copied. Again, the structure of DNA is these nucleotides, and this is the chemical structure of nucleotides. But basically, the core concept is this is a C pairing with a G. There's 3 hydrogen bonds. And this is a T pairing with an A, where there's 2 hydrogen bonds. So again, the concept of sequencing is we can split these 2 strands apart and copy one of the strands so that we can understand the sequence of that DNA that we're looking at. So what's really important in the concept of sequencing is this 3' hydroxyl group. So the structure of a nucleotide, you have this sugar group, you have this base, which is the ACT or G, and then you have a 5' phosphate group and a 3' hydroxyl group. This 3' hydroxyl group is important for copying DNA because when a nucleotide comes in, it undergoes a nucleophilic attack of the incoming phosphate group, and it creates a phosphodiester bond. So again, you'll get all of this again in subsequent lectures, but just kind of understanding these concepts at the base level is important. So what we realized is that if we created a synthetic nucleotide molecule that lacks that 3' hydroxyl group, which is called a dideoxy DNTP, because the dideoxy, meaning there's no hydrogen oxygen group at both the 2' and the 3' position. If it lacks that, then it can no longer extend. It can't incorporate the next nucleotide. So it creates a stop in the sequence. So what we realized is that if we created these synthetic dideoxynucleotide molecules that were fluorescently labeled a certain color, we could detect the identity of a nucleotide at a certain position. And that's the concept of sequencing. So this picture is kind of hard to see, and I don't expect you to know like every detail of this, but this is the basic framework of Sanger sequencing. So essentially what you do is you incorporate these dideoxynucleotides that lack this 3' hydroxyl group, and it creates what's called chain termination. So if you do this in PCR cycling, you create these fragments that are representative of each base at each position of the DNA. And then you can separate those via capillary electrophoresis and create what's called a chromatogram or an electropherogram, which is this series of peaks that give you a different color telling you the nucleotide at each position. And it's as basic as that. And we can look at these and say, okay, this color corresponds to a T, this color corresponds to a G, this one a C, this one to a T, and then you can just kind of sequence A, C, G, T, A, C, A. And that's the basic concept of sequencing. Is that okay so far? Clear? Yes. So the evolution of sequencing, and probably back in, I don't know, like the late 90s, early 2000s, what we would do is we would create these terminated fragments and we'd run them on an agarose gel. So I think you actually go into the lab and you do some of this kind of stuff where you run out TCR products on a gel. I'm not sure, but I think you do that. If not, we'll hopefully incorporate that into the program at some point. But it's the same concept where you have all of these fragments that correspond to the size and the position of the nucleotide at that size. And in each lane, you run the terminated fragments for a specific nucleotide. So all of your fragments that end in A, you run in lane 1. All of the ones that end in G, you lane in run 2, lane 2, and then lane 3, lane 4, etc. These migrate based on their size, so that the smallest fragments are at the bottom and the largest are at the top. So now you can sequence. You can see the first base is a C, second base is an A, the third base is a G, the fourth base is a C. So you kind of see how you can sequence that strand of DNA via this process. Is everybody seeing that? Okay, cool. So again, Sanger sequencing basically enabled us to use capillary electrophoresis and fluorescence so that we could generate these chromatograms. So same concept. Again, you can see, C, A, C, G, G. Similar concept as a gel, it's just more automated, and it gives you a data output that is more efficient. So we could get faster and sequence in a day, as opposed to these gel techniques took like 3 or 4 days to do. So now the concept of next generation sequencing, where you're massively parallel sequencing many genes at a time, the concept of bioinformatics came into play. And this is what data looks like for next generation sequencing. And basically what this is you have a reference chromosome at the top, and then all of these are reads, or how many times you've sequenced a particular base. And anytime that you see a difference in color, or this black line represents a deletion, you can see changes in the DNA. So if it's like just solid red or solid blue, then there's no changes. It's the same as the reference genome. But once you see these colors coming in here, that means that there's some kind of mutation or variant that's happening that might be important for that particular individual. It could be a biomarker for cancer or some genetic determination that they have something mutated in their DNA that is causing their disease or their condition. So this is kind of what happened that enabled us to apply these types of techniques. So what you're looking at here, as this is going up, this is the amount of data that we could generate in a short period of time. And this is the cost that it took to generate this data. So the sweet spot was when these two came together. So, back when we were doing things via gel electrophoresis, it cost like $10 million to sequence the whole genome. Once we got to Sanger, it got a little less expensive. And now with next generation sequencing, we can sequence the entire genome for like $1,000. So really, this is kind of the time in which this became applicable for doing this in a clinical setting around 2014. So this is like a mental break slide. Cats taking selfies. I do have small children, so apologize for my rudimentary humor. All right, enough of that. Okay. So now we're going to go into next generation sequencing. We have a sort of core understanding of what sequencing is. Next generation sequencing, it seems complicated and overwhelming at first, but if you break it down, it's established off of these core concepts that you can isolate DNA, you can break it apart, you can copy it, and you can look at it. And next generation sequencing enables us to look at it bioinformatically, so we can look at data and use bioinformatic pipelines to determine where changes occur in the DNA. And that gives us information about the patient. So this is just a framework of what the instrument looks like and what the consumables look like if you ever do this in the laboratory. So the instrument is like the size of a microwave. It's pretty small. It would fit kind of on this table here. It's like this big. And you have these cartridges that you load sample from a patient into the cartridge, and you mix it with enzymes and buffers, and you put it all in this instrument, and it takes about 36 hours, and then it gives you the whole genome sequence of that individual. So this slide kind of just goes through in text format the basics of next generation sequencing. So NGS is massively parallel DNA sequencing, meaning that we can sequence many genes at once. The starting material can be DNA or RNA. That's a little bit of an advanced concept. What we found is that you can sequence the DNA and you can determine the content of all the genes. But sometimes what happens is after DNA is copied and it gets transcribed into RNA in the cell, that RNA becomes protein. There's processing steps that change the configuration of the RNA. So in some cases, you have to look at the RNA to really know what's going on. So in the lab, what we're doing is sequencing both the DNA and the RNA, and that gives us a clearer picture of what's going on with the patient. And the sample types essentially can be tissue, cells, blood, or other bodily fluids. So we can sequence from saliva, from buccal swabs, and other things that we can isolate from patients. Anything that has DNA in it, we can sequence. So you can sequence the whole genome or just the whole exome. Does anyone know the difference between genome and exome? So DNA is made up of exons and introns, and the exons are all the protein coding content, and then you have a whole bunch of repetitive sequence that are contained within introns. Those introns get spliced out in a process called splicing, RNA splicing. And ultimately, the exons are what go on to encode the proteins. So in a clinical setting, sometimes we're just interested in those exons. And the reason for that is a couple things. One is the exons gives you most of the information about what is actually happening biologically in the cell. And two, the exon content is about 2% of the total DNA. So instead of sequencing 3 billion bases, we're only sequencing 2% of that. So you can see how the cost can be tremendously lower if you're just looking at the exon content. So that has a lot of application in the clinical setting, and that's typically what we do for patients, sequence their exome. And then we can also sequence just targeted gene panels, and that's what we do a lot for cancer, where we know there's like 15 or 20 genes that are involved in a certain kind of leukemia or lymphoma or whatever the carcinoma is. And we can just look at those particular genes, and that keeps your costs down. Instead of like $1,000 for an exome, we can sequence panels for like $200 or $300. So one of the key sort of concepts of next generation sequencing is it involves linking DNA adapters to the genetic information. So you isolate the DNA from an individual, you fragment it up into sizes that you can copy and sequence, and you connect these DNA linkers. And those are synthetic pieces of DNA that enable you to capture the molecules on a solid platform. And then you can use enzymes to copy those molecules. So the concept of DNA adapters is important to sort of understand next generation sequencing. Another concept is that NGS is digital. So the output is in a digital format. So it enables us to use bioinformatics. It's kind of in that binary 0 or 1 format where it's either an A or a T or a C or a G. So since it's digital in a sense, we can quantitate. We can tell how much of a certain gene is present within an individual and how much of that population is mutated or variant. And that can tell us how sick a person is or if they qualify for certain therapies and things like that. So the advantages of next generation sequencing versus Sanger is it's higher throughput. Again, multiplex, you can sequence a bunch at a time. And the cost per gene is low. So, like I said, Sanger took $10 million to sequence the genome. Now we can do it for $1,000. So the cost per gene is much lower. And the sensitivity is better since it's digital and it's quantitative. We can detect 5% variant allele frequency, which is important in cancer because tumors have a very low percentage of mutation. So we can go down and detect really low levels of mutated DNA. And you can't do that with Sanger sequencing where the sensitivity is like 20%. So NGS has a much higher resolution. So some of the disadvantages of NGS, we can only sequence very short fragments. In Sanger sequencing, you can sequence a kilobase. In next generation sequencing, the fragments are about 300 base pairs. Again, it's a pretty advanced concept, not something that you have to remember for an exam. But that's kind of a big disadvantage of NGS is we're looking at short fragments. An advantage of that, though, is we can sequence from tissue isolates. So what we do when we do a biopsy or a resection in a cancer patient is we store the tissue in formalin-fixed, paraffin-embedded, which is basically wax. And when you wax the tissue, it fragments the DNA. So you have to use short read technology to sort of piece together the sequence of DNA from those samples. So NGS can do that, as where Sanger will sequence much longer fragments of DNA, which has some advantages with structural things like that. But in general, Sanger is for longer reads, NGS is for shorter reads. Next generation sequencing has complex analysis. Bioinformatics is hard. A lot of our bioinformaticians have PhDs in data science, so it's a pretty intense discipline where you have to, there's a lot of education and training to be able to do this kind of stuff, but everyone's capable of doing it. You just have to go through the training and education to learn how to do it. The overall cost of next generation sequencing is high. And I know that's a little counterintuitive to what I said here, but we're sequencing a lot more genes. So again, it costs us like $1,000 to sequence an exome. And Sanger sequencing, if you're just looking at one gene, it's only like $200. But again, in NGS, you can look at 20,000 genes. So the cost per gene is lower, but the overall cost of the test is higher with next generation sequencing. And we think about that as a clinical lab because we don't want our tests to be too expensive because then patients can't afford it. So this is like the basics of an NGS workflow. So again, you start with genomic DNA, you fragment that genomic DNA, and then you add these DNA linkers. So these adapter sequences. So the black bar in the middle is the human genomic DNA. And then these colored bars on the end are those adapter sequences that enable you to capture those fragments and then sequence them on the platform. So what happens then is you capture these fragments on what's called a flow cell, which is basically just a solid piece of silicon cartridge or plate. And then once those are captured, you can make a bunch of copies. So that concept of copying DNA, going back to that core concept, you make a bunch of copies. These are called clusters. Each cluster represents an individual molecule of DNA. So then you can sequence each of those clusters basically by incorporating different colored nucleotides. So going back to the core concept, those dideoxynucleotides that are different colors, and you can determine the nucleotide identity of each of those clusters or the sequence. And then ultimately you get these reads, which tell you this gene has this particular sequence. So the data output, or what this looks like, is you get these reads of sequence. And these sequence align on one another, and then it can tell you if there's a mutation at a certain position. So in this case, at this particular position, in the reference genome, a healthy individual, they have a C at that position. But in this particular fragment, it's saying that C is mutated to a T. So that could be a biomarker for cancer or something like that. So next generation sequencing enables us to determine all of those mutated positions within the genome. And then again, it's digital, so we can quantitate that, so we can tell at what percentage is that represented. So in this case, if you do simple counting here, the math adds up to about 86% variant allele fraction. So that meaning that the C is present in about 86% of sequences, and the mutant is present in whatever 14% of sequences. So again, this is what your data output actually looks like from a bioinformatician perspective, where you have these reads that line up. If they're all gray, it means there's no mutation. If you see color, you guys can see the color there. I know these slides are kind of hard to see. But that tells you that there's a variant at that position, and that's something that we want to investigate and interpret because it probably has clinical utility. And that's the concept of next generation sequencing. So we can also multiplex in next generation sequencing. What that means is we can sequence multiple patients at one time. And the way we can do that is leveraging those adapter sequences that I talked about. So again, the concept of this is if you have genomic information from patient 1 and genomic information from patient 2, you can put different linker sequences on patient 1's genome versus patient 2's genome. Then you can pull them all together and you can sequence them, and then you can demultiplex based on that linker sequence. So you can see patient 1 has this blue sequence, CATT-CG. Patient 2 has this purple sequence, AACTGA. So now you can separate patient 1 versus patient 2. And that just enables you to sequence many patients at one time. So depending on the platform, we can sequence like 20 patients in a single run. So in 36 hours, we can get 20 genomes. So it's pretty powerful technology if you think about it. So these are some of the platforms that are available for doing this type of thing in the laboratory. So Illumina is my favorite chemistry and what I've used my entire career, but there's others that are just as good and they have similar characteristics. But I'm not going to go into too much detail here other than say Illumina uses this sequencing by synthesis chemistry, which basically just means that you incorporate fluorescent nucleotides, and then based on the color of the nucleotide, you can determine the sequence. That's all there is to it. That's simple, right? So Ion Torrent uses this semiconductor pH sensing chemistry. And what this is based on is when you incorporate a nucleotide, you remember that hydrogen at the 3' position that I talked about undergoes a nucleophilic attack of the incoming nucleotide. What happens in that chemical reaction is you release a hydrogen, and that can be measured because that causes a change in the pH. So in this chemistry, you capture the DNA in this little flow cell, and then you add nucleotides. And if the incoming nucleotide matches the template strand, so in this case, the template strand is a G, so a C is going to come in base pair. So when the C comes in, it releases this hydrogen molecule, which causes a change in pH. So it tells you there's a C at that position, or in this case, sorry, a G at that position. So then you get a sequence. So just kind of understanding at the basic level, this is using fluorescent nucleotides to measure or tell you the nucleotide sequence. This measures change in pH to tell you the base identity at each position, but these are just different ways to sequence. So now they have these 3rd generation sequencing platforms. So it gets even more complicated. Sorry, this thing's falling apart on me. I need to get a new laser pointer. I'm not even really using it, just holding it. So there's 2 kinds of 3rd generation sequencers. And the reason that these are called 3rd generation sequencers is because the difference between 2nd and 3rd is in 2nd, you make a bunch of copies of the DNA and then you sequence it. In 3rd generation, this new chemistry enables you to go in and sequence the molecules directly. So you can just isolate DNA from an individual, put it on these platforms, and you can directly sequence those molecules. The advantage of that is you don't have to synthetically copy the DNA. In the process of synthetically copying DNA, you can the enzymes make a lot of mistakes. So you can get these false positives or variants that aren't really real. They're a result of the amplification chemistry. In third generation sequencing, you don't have to make all those copies. You can just directly sequence the DNA molecule. And there's two versions of this. One, PacBio, it has this enzyme at the bottom of a little well, and it captures the DNA. And then you incorporate different colored fluorescent bases as the DNA moves through this protein channel. So you can tell as the DNA is moving through if there's an A, C, T, or G at that position, and ultimately sequence the DNA. So Oxford Nanopore is another one. This is based on ion channeling. So basically they have an artificial ion channel that you feed the DNA through. And as it's getting fed through the channel, you blast it with ions, and those ions move at a rate that's relative to the nucleotide at that position. So in A, the ions will move at a certain speed. C, they'll move at a different speed. At G, they'll move at a different speed, etc. So as you move the DNA through this pore, you, in a sense, sequence each position of the DNA as it's moving through. And that's Oxford Nanopore direct molecule sequencing. Again, these are advanced concepts, but cool stuff. And if you do get into the lab and in the molecular world, you'll start to interact with this type of technology. So it's pretty fascinating. So the advantages of third generation sequencing is you're directly sequencing DNA or RNA. The disadvantage there is you need more starting material because you're not copying it. So you need to isolate a higher quantity of material. That's challenging in the pediatric world because we typically only get a little bit of blood from a baby. So it's hard to do this yet in pediatrics, but in the adult population, they're starting to do it more because you can get more sample from an adult. You can do long reads, so it enables you to do over 15 kilobases, but it has a higher error rate. The error rate's 10 to 20%. In the second generation sequencing, the error rate's only like 5%, so you get a little bit more error. But bioinformatics helps you tease that out. And it requires assembly and alignment, and it can be expensive. So these are just some of the sort of the core advantages, disadvantages. So this is an overview of what bioinformatics looks like. So again, this is your sequencer. You have data files that come off the sequencer, which basically packages all of this information. So again, sequencing 20,000 genes, each of those genes is going to have a different nucleotide sequence. And you're going to use bioinformatics to kind of figure out the identity of all of the genes that you're sequencing within that sample. So this is done via bioinformatics. So bioinformatics, the concept of it is very simple, and then it gets complicated the more into the weeds you get. But basically all it is you start with this huge file of all of this genetic information, and you filter it down or distill it down to just the stuff that's relevant for clinical application. And you do that via bioinformatics. So you're basically getting rid of all of the stuff that doesn't mean anything. And you're just focusing in on those mutations or those variants that you then interpret into a clinical report that enables you to care for a patient. So I'll go through this quick because it's really just core concept. You get these big files, and this is called the primary analysis. These are like a terabyte of data, which is a lot of data. And in primary analysis, you align those fragments of DNA to a reference genome. And it's like putting a puzzle together. So you have the picture on the box, and then you put those pieces together based on that picture. And that's what alignment is. And again, what you're doing is you're looking at each nucleotide position and determining if there's any mutations. In this case, C is the healthy individual. And if they have a T at that position, then you know there's a mutation there, and you can go in and interpret and see what that means. In secondary analysis, again, you're just distilling this data down. So you're applying all of these filters to take a huge data set and make it into a small data set. So you get rid of the junk, all the poor quality sequence. You get rid of polymorphisms, which are variants that exist in the natural population. So all of us have some variant in sequence that is just normal and healthy. So you really just want to zero in on the clinically relevant stuff. And then you filter out the false positives and false negatives, and essentially this becomes your bioinformatic pipeline. So now after secondary analysis, you have a variant call file that just has those clinically relevant mutations or variants. And those are what go on to get interpreted or annotated and interpreted into a clinical report. So this is where it hits my desk as a medical director. I get the data in this format after it's already gone through all of this bioinformatics. these are my bioinformaticians, the data guys that do all this. And then they give me this file and they say, now you go in and interpret this stuff. These are the mutations that exist in this individual. So I use databases like ClinGen, Cosmic. There's tools that are available. If you ever get a chance, just Google ClinGen or Cosmic and you can go and look at the database. What that is a catalog of all of the mutations or variants that are known to have implications in disease and cancer and stuff like that. So if I say, well, I have a T at this position at this variant allele frequency, I blast it against this database and it says, that means this person qualifies for EGFR mutational therapy. And then we can go on and prescribe medicine, specific to that individual's genome portfolio. So ultimately what we do is we give the doc a report, and then they can look at that report and to see all of the clinically actionable mutations or variants, what medicine the patient qualifies for, if there's any clinical trials that are open that they qualify for, and then that helps how we determine the disposition of the patient. All right, we'll do one more mental break. So dogs are not to be overdone by cats, so they had to get their supplements in there too. Again, I have an 8-year-old and a 4-year-old, so they help me with this slide. Okay, so this last concept is optical genome mapping. I'm not going to spend a lot of time on this because this is like the newest, latest, and greatest technology that we're using in the lab. I apologize. I should have mentioned before, these things are abbreviated. So next generation sequencing, people mainly just say NGS for next generation sequencing. For optical genome mapping, we're lazy, so we just call it OGM instead of saying optical genome. So if I say OGM or NGS, just that's what I mean by it. Optical genome mapping is really cool. It uses an instrument, again, it's like pretty small, the size of a microwave. That's relevant because in a clinical lab, we can't have these huge instruments. They have to be benchtop instruments. We don't have a lot of space and resources. So anytime we can get this down into a small instrument that we can put on the bench and we can use it for this kind of stuff, that really enables clinical application. So you got this instrument, and what it does is all of your sort of classic cytogenetic techniques. So if you guys have any background in cytogenetics, those techniques are like karyotyping, microarray, FISH. It's really where you're looking at the structure of chromosomes, because mutations happen at 2 levels. In sequencing, you're looking at the base level. So 3 billion bases, you're looking at the identity of each of those bases. In cytogenetics, you're looking at the chromosome level. So chromosomes have these, they make mistakes as they're being replicated where they break apart and they undergo things like translocations or homologous recombination, and it creates diversity. So a lot of us we're all very similar in terms of our DNA sequence, but what makes us different are those sort of changes that happen with our chromosomes via recombination, chromosomes breaking apart and coming together in different ways, and that is what makes us unique as individuals. So this enables us to do this in a digital format. So I really like digital technology because I'm into data and I geek out on data science. So anything that can give me a digital readout, I can interface that data into bioinformatics and in systems like Epic and laboratory information management systems, so that the data can integrate into the patient medical record. So that's really where we're going in the world of clinical testing and clinical pathology, is moving to these tools that are more digital readout as opposed to like things that you have to look at and interpret. It gives me data that I can then put into databases and figure out what mutations mean and how to interpret them. So optical genome mapping is great because it takes that cytogenetic type stuff and it puts it into a digital format. So where I can use similar techniques like next generation sequencing to analyze data and interpret data. So this is a kind of breakdown of this workflow. So again, just like next generation sequencing, you have to first isolate DNA from the individual. And this, again, is represented in chromosomes because we're really looking at these big structural things that are happening in the chromosome. And then you label each of those fragments of DNA. And DNA has replicative sequence like every 500 base pairs. So what we found out with this technology is we can fluorescently label every replicative sequence in the DNA. So every 500 base pairs or so, you get a label. And then based on the series or the sequence of those labels, you can determine if there's changes that are happening in the chromosome. So then you put it on the instrument, it gets those fluorescent molecules get linearized and put through the analyzer. And that's where you see these kind of like starry night photos, which I think are really cool. Like when you first look at them, it just looks like you're looking at a sky full of stars. And it doesn't make much sense until you do the bioinformatics piece of it. So in the bioinformatics piece, you use algorithms to convert that starry picture into DNA molecules. And what you see here is each of these molecules have these fluorescent labels at that repetitive sequence. So then you can align those molecules to create what's called a consensus sequence. So now from that information or that nucleic acid that you isolated from the patient, you see the series or pattern of fluorescently labeled molecules within the DNA. And then you can compare that pattern of fluorescent molecules to a healthy individual or a reference genome, and you can find all of the changes or mutations that are happening within that individual sequence. So this is the data output of OGM, which is kind of cool too. You get this sort of spider wheel. And this tells you all of the big sort of structural changes that are happening in the chromosome. So these are the types of structural variants that you can see with OGM technology. So deletions happens when a big fragment of DNA is deleted. And you can kind of look at the patterns. And this is showing you the green is the reference genome and the blue is the patient. So in this case, the reference genome has 1, 2, 3, 4 fluorescent bands, but the patient only has 1, 2 fluorescent bands in that region. So it tells you that something has been deleted from the patient's DNA. So that's kind of the concept of all of these data packages. So you can see insertions where you go from small to bigger. That's not me. There's red on here, and I was like, am I bleeding? I'm not. I don't know what that is. Probably from a sharp, one of these markers. Repetitive, so you can look at tandem duplications and DNA being repeated in the sequence. And then you can also look at what are called translocations, and that's when a piece of DNA breaks from a chromosome and gets moved to a different chromosome. So you can see all of this type of data in mutation patterns within optical genome mapping. Yeah, sorry.
So with this, can you see specific genes or specific areas?
Specific areas, that's
Okay.
a good question. So the resolution is about 500 base pairs, so some genes are smaller than that. So, but a lot of these types of structural changes involve many genes. Like most of them involve like 20 to 50 different genes. So ultimately, when we get a readout, it tells us all the genes that are involved. And then we look at those genes and say, is there a clinical consequence of this gene being deleted? So it's a good question. Yeah, you typically see some number of genes, like 10 to 20 in that range. If you want to look at like the individual gene level, that's where really we start thinking about next generation sequencing. So this is just showing you like what these changes look like on the chromosome. So deletions is like this fragment's getting deleted. So you see it here, but not here. Duplication, it gets copied. So here you have 2, here you have 4, it gets repeated. Translocation are like different pieces breaking off and rearranging onto different chromosomes. And inversion happens when you have a sequence of DNA that flips. So it goes from left to right to left. Yeah. So this is your inversion. You can kind of see it flipping. The pattern goes from this direction to flipping to that direction. Okay, good. So a key concept of OGM is it replaces classic cytogenetic techniques in the laboratory. So karyotyping fish in microarray, again, you're going to have these slides so you can look through all of this. I think it's more fun to look at the pictures to understand the concept. So karyotyping is just looking at all the chromosomes. And cytogeneticists that have a really trained eye can look at this and tell you where the difference is. Can anybody like tell me what's going wrong with this? It's a very common genetic disorder. Yeah, good job. Trisomy 21. There's an extra copy of chromosome 21. So you guys are pretty good. So in fish, you can look at the gene level if genes are copied or translocated. So in this case, this is a normal isolate. So they have 2 copies of each gene. If there were 3 copies, you would see 3 of a certain color instead of 2. So that tells us if there's extra copies in the cells of individuals. And then microarray is a digital technique for looking at duplications and deletions. So you get a digital readout, and if you see something going down, then that means it's being deleted. If you see it going up, it means it's being duplicated. So we can look at the whole genome, and we can determine where all the duplications and deletions are, and if those have any clinical consequence for the patient. So OGM does all this in one technology. So instead of doing 3 different things that take 2 weeks to do, in 3 days, we can do OGM and get all this information. So it's pretty cool stuff. So what my vision is, and again, you won't be tested on this. They don't care about my vision on your test. But what I want to do at Akron Children's Hospital is have both NGS and OGM. Because to your question, NGS enables us to look at that small stuff. And then OGM enables us to look at all the big stuff. So now we can do 360 genomics. We can see all of the mutations and variations and aberrations that are happening in a human genome. And that enables us to make the most accurate diagnoses for a particular patient. All right, a few slides on application, and these I'll go through quick. So this is fun. It's a case study. This is an actual patient that we saw. So this is a male. They were in their 30s, had some colon stuff going on. Primary, meaning they had some kind of brain tumor. You can see in the imaging the tumor, this guy here. That's not normal. You want that. And it also metastasized to the spinal, so you get some metastatic things in the spinal. If you see this little white area here, that's a tumor. So the first thing we do is look at this histology. So we look at this in the laboratory. An anatomic pathologist looks at the tissue. If it's stained, the more purple it stains, the more indicative it is of cancer. So you can see this individual has some cancer. Next generation sequencing tells us the complexity of the genome. So what we saw here is on primary, they had a fairly complex genome. When they went metastatic, the level of complexity increased pretty dramatically. So these fish plots down here, the more color, the more complex it is. And what that's cool is that complexity of the genome is a biomarker for certain chemotherapies. So at their metastatic phase here, when we tested this, we realized highly complex genome. So we switched them from TMZ chemo to pembrolizumab, which is a immunotherapy. So a complex genome is indicative or an implication for immunotherapy, which basically takes that complex, highly mutated genome and corrects it and gets it down to the healthy genomes in that individual. So then this person basically, after we started them on Pembro, they went into complete remission. So what you see here is immune function on primary metastasis. We didn't see any immune functionality, so the brown is immune cells. So after we started them on Pembro, their immune function went into overdrive, and now you see all this brown stain, which is immune functionality, and the patient went into remission. So this is just a case study of how we use all these technologies, pair them together to try to get patients into remission in the case of cancer. For genetic disorders, we're looking at, we use this technology for intellectual disability. So as a pediatric hospital, we get a lot of autism and other types of intellectual disability that we try to diagnose. Rare disease, like we said, things like trisomy 21, and there's a lot of disorders and things like that we can detect, bless you. And then also we use it for reproductive health. So we can use it in the maternal fetal medicine setting. If there's complications with pregnancy, we can use genomic technology to determine what's going on and why those complications are existing. Pharmacogenomic testing, again, I kind of introduced this before, but the basic concept of this is to apply genomics to determine how to dose a patient. So you determine how they metabolize medicine, and based on their level of metabolizing it, you either up or decrease the dose of medicine. And this just tells you it's different if the drug is active versus inactive. Again, you have this slide. This won't be on the test, I don't think. I mean, pharmacogenomics is pretty advanced. But, it's kind of cool to think about how we apply this stuff. So if the drug itself is active and you're not very good at metabolizing it, then you want to give the patient less drug because it'll become overactive because you're not breaking it down fast enough. So you can have cytotoxic effects of the drug dose. As in the case where if the convergent state of the drug, so your metabolism needs to break it down for the drug to work. If you're poor metabolizer, you need more drug because it takes your body a longer time to break it down. So giving you more drug, you'll see the effects of the medicine. So that's the concept of pharmacogenomics. And then the last slide here is just in the infectious setting. And this was big during COVID. I guess shameless plug. In my previous lab, we were one of the first labs to sequence SARS-CoV-2 using this technology. And now a bunch of labs do it. It's old news. But basically what that enables us to do was determine the variant profile that was circulating in the population based on the genomics of SARS-CoV-2. And then that helped us understand, like, how severe people were in terms of their illness when we had different phases of infection circulating through the country and the world. And it also enabled us to break this down into phylogeny. So we kind of understood where those variants came from. And based on the family tree, we could tell like this variant is a little bit more severe than this one. So these patients tend to get a little bit more sick if they have Delta versus Omicron. So that was kind of how we applied genomics in the area of infectious testing. And viral genotyping is a big application. You get it with things like HIV and HCV, HSV, where we can genotype the virus, and that helps us better understand how to treat that particular patient. All right, cool. So we're right on time. We'll do a quick quiz together, and then we'll wrap it up. So what's the difference between Sanger and NGS? That's right. In NGS, you can sequence many genes at a time. Sanger is only one gene at a time. That might be on the test. So what classic cytogenetic techniques does OGM replace? Karyotyping, FISH, microarray. All right, you guys understood everything I said today. Okay, so bonus question. And this is the type of math that we have to do in the clinical lab every day. So you have a patient, they have a heterozygous variant present in 50% of tumor cells. The tumor cellularity is 40%. What is your required limit of detection to pick up that variant? Remember, it's heterozygous, not homozygous. Any guesses? We'll go through it together. Tumor cellularity is 40%. It's present in 50% of tumor cells. What's 50% of 40? 20%. So it's heterozygous. So if it's heterozygous, remember there's two copies of each gene. So if it's homozygous, both copies have it. If it's heterozygous, only one of the two has it. So what does that 20% come if it's a heterozygous? 10%. All right, so that's the kind of math that we have to go through to determine the limitations of our testing. If we want to be able to detect, and it's kind of challenging at first because you're like, well, this patient has 50% mutation in their tumor cells, but you have to take into consideration that how many of those cells are tumor versus how many are normal. And then if it's a heterozygous variant, then it's only half. So you got to divide again by 2. So in this case, we need 10% sensitivity to be able to detect this type of variant. NGS can go down to like 5%. So this is a typical, we can detect something like this in the laboratory. Okay, any questions? So you have these slides. Again, this stuff is kind of rich, but I don't know if you have my email, but you can always stop by upstairs in the hallway. My name's on my door. So if you are interested in getting involved in doing this kind of stuff and you want to chat about it, just stop by and let me know. You're welcome. I went to Mount Union, by the way. I see a Mount Union. So long time ago, but